Hi everyone, my name is Kristyna Kupkova and I'm going to introduce you to our new package called GenomicDistributions, which is basically this rich toolbox of functions to analyze, visualize, and to compare genomic intervals. So basically any bed file that you might have. We can go to the next slide. Why should you be interested in genomic distributions, and why should you use it? I can give you 4 reasons. First, as I said, it's a really rich tool of functions to describe genomic regions. So we really put it any descriptive function that we could think of into this package. So everything is at one place. It's really easy to use, and you don't have to go anywhere else. Second, we wrote a package in a modular way where all of the functions are separated into calc functions that pre-processes your data and plot functions that does the plotting. So once you have your input, you first run it through the calc function and you get your distribution or your statistics in form of a table or a list, which you can then easily plot yourself. This kind of offers you more flexibility. Or if you want to do kind of quick and dirty analysis first, you can plug in the output into our plot function, which then provides you a ggplot object, which you can of course then further edit also. So this kind of gives you this like double degree of flexibility. Furthermore, all of the functions are taking either single query input in form of genomic ranges list, or if you want to process multiple queries at one time, you just provide a genomic ranges list to the function, and everything is processed for you in one single step. So you don't have to worry about writing a for loops or about using apply functions, etc. So it's really easy. Furthermore, we really put an effort into writing the code, and everything is optimized for speed. So all of the calculations are done really fast. So if you have large data sets, I really do encourage you to use genomic distributions. If we can go to the next slide. Here I have a few examples of what genomic distributions can do. So if you have your regions of interest, you can plot their distributions across different chromosomes. You can find if your regions are enriched for open chromatin signal in a given cell type. You can find out distances from certain features, for example, transcription start site or any feature that you might provide. You can find out if your regions are in GC rich or GC poor regions of genome. You can find the distances between individual intervals, or if your genomic regions are enriched in certain partitions. So are they mostly in exons or introns or etc. With this, I would like to encourage you to use genomic distributions. I provided here a link to our GitHub repository. Who missed that, I provide the link in the poster that I presented yesterday. It's called Genomic Distribution, so you can check that. And if we can go to the next slide with this, I would like to thank all the people who contributed to making this tool, to making this package, and who supported me through my PhD. Thank you for your attention, and I guess this would be all for me.
Great, thanks very much, Kristyna. This was very, very nice. I just would like to remind the participants again how this is working. So we are playing the slides here. Each of the speakers in this session has 4 minutes to present. You can post your questions to the conference platform under the polls. And at the end of the session, we'll have a Q&A together with all speakers. All right, so now the next speaker, and I hope that I pronounce your name the correct way, is Sangram Sahu.
Yes, yes, that's correct.
Can you see your slide?
Yeah, I can see the slides. Thank you.
All right. So thanks for joining, and you are very welcome to start.
Yep, thank you. So, hi guys, my name is Sangram Sahu. So, I'm currently based out of Bangalore, India. And this SigBio-Shiny is a kind of like my pet project, I can say. I do out of my free time. So, yeah, so this is a basically a Shiny application wrapped in a R package. So, what it does basically, like you can do most of the stuff with a set of genes basically. Yeah, so today's outlines will be like basically what a gene set means and what are the available resources and some of the ideal scenarios to doing so. And we'll see SigBio's overview, its features and small demo, and like how I'm trying to collaborate with modular design. Next slide, please. Yeah, so basically a gene set, after any kind of analysis, like suppose for a typical transcriptomic analysis, you get a set of differentially expressed genes, up and down regulated genes. So this can be further taken into different kind of enrichment tests like SCGA, ORA, or NTA, right? So, and they give you meaningful biology going further with those set of genes, correct? So yeah, so basically this is what this application trying to do. Next slide, please. Yeah, so there are some great resources already available, like your MSigDB GSEA or Enrichar or gProfiler or David, but they have like slight problems with them in some key scenarios. Next slide, please. Yeah, so if we consider a ideal scenario, so basically for any kind of thing, we need an up-to-date resources, right? Like the up-to-date database, which have all the IDs mapping and everything, right? And sometime like with passing time, we require this for non-model organisms as well. So most of the databases, whatever they have for model organisms, and also we need to keep in mind of the reproducibility thing, and that can be achieved keeping your database version same and statistical the iterations, number of iterations you do while doing the enrichment, all these things, keeping a log of them. And apart from like GUI which is provided by David and everything, you need a programmatic access as well, and some direct end or enriched plots, right? So yeah, like some resources have some of the components but not all. So yeah, that's what we're trying to achieve in here. Next slide, please. Yeah. So this SigBio-Shiny is like completely depend upon bioconductors open infrastructure resources. So it's basically using annotation hub to getting the different IDs from different databases, and then using some of the bioconductor packages on top of those resources to do the analysis and everything. Yep. Next slide, please. Yeah. So this is how what things in background goes. So initially, this can be launched from directly from a Shiny server, or it can be used launched from your R console. With if you have a set of gene list, or along with gene list and your fold chains, you can directly launch that. So and you need to provide your organism details. So it will fetch the organism database in real time, so that you can make sure you getting your updated database, and it convert those IDs and everything. Then there are different modules. So in different modules, different kind of enrichment test happens. Next slide. Yeah, so these are the all things like it's currently doing: downloading database, doing gene mapping, ontology enrichment, and pathway enrichment, and few things are in work in progress, like creating a reproducible R script, so that at any point if you run, you make sure like your database is same, all the things whatever you've done previously through the GUI, those stay same. So it helps in reproducibility.
And that's time.
Okay. Yeah, I don't have much actually. Yep, thank you.
Do you want like 1 minute more or so, or do you want to just perhaps wrap up your talk, or say some final words?
Yeah, just next slide. When I can show them. Yeah, so this is how it can be loaded. One more slide. I can show the just GUI. It's a basic GUI. Okay, I'll give you the link, like maybe I'll say in the chart like how this works and everything. Yeah, that's it. So can you go to the last slide? I just want to provide the links. Yeah, so currently I am totally developing independently. So from here, like my idea is to, if I can get someone on board, we can collaborate, then we can make it into a stable version, much stable version. Yeah, so that's it. Thank you guys.
Thank you very much, Sangram, for this very interesting talk. Yes, just it would be wonderful if you can post this links to the platform so people can join you and perhaps collaborate together. Thanks very much.
Sure.
And again, you can post your questions to the speakers on the conference platform under polls. And it helps us also if you add the name of the speaker, say, whom is your question addressed to, because we have 7 speakers. So it's easier than for the Q&A session at the end. Right, the next speaker is Nathan.
Great. Okay, I wanted to just take a couple minutes to share with you a project that we've been working on in my group called Refgenie. So this is a tool which is designed to try to solve this problem that many of us have faced for many years, and that is that many tools require genome-related assets like bow tie indexes or BWA indexes or annotations that relate to a genome, but we typically store these assets differently. So, I mean, we have a different folder structure, different labs, different individuals within a lab may store these differently, or different labs may store them differently, which leads to challenges with interoperability, because if we're writing software that needs these kinds of assets, then we can't get to it exactly because they don't follow the same structure. So next slide, please. So if we had a standard organization, then you could pass to a pipeline just the name of the genome, and it would sort of know where to find the different things it needed, instead of, which I'm showing sort of on the top line here, instead of the alternative, which is the second one, where we would need to pass each thing independently because they sort of might be in different places in different people's environments. So that's the value of a standard organization. So next slide, please. So one solution to this is the iGenomes project. If you're not familiar with it, you can go check it out. Illumina has basically produced standard resources in a tarball that you can download, and then they all follow the same format. And now we can sort of all use one structure, and you just say, give me the iGenomes, and then my stuff will work. Next slide. But the problem with that is that it's not scripted. So if you want to use a genome that they don't have, well, you're out of luck. You could try to recreate it, but that would be kind of tough. It's not modular, so you've got to download the entire thing, which is everything they provide. You can't just get sort of the one thing you need. All I needed was the bow tie index, for example. And it's not programmatic. There's no way to sort of explore these via an API or anything. It's basically just a tarball that you download. So next slide. So Refgenie is an attempt to solve these limitations. It's a command line tool that provides 2 ways to retrieve these assets. You can build the asset for any genome of interest. So everything's scripted, unlike iGenomes. So that means I can recreate this environment for my spike-in genome or my non-model organism or my personal genome or whatever it is. But in addition, there's also a pull function, so we can just pull stuff if the asset happens to already be built on a server. It also provides better discoverability in that there's an API from this Refgenie server instance, which provides a browsable web interface and API to look at the different assets. And then the client allows us to obtain the local paths of the asset really easily with this seek function. Next slide, please. So this is a simple example of how it would work on a command line. You just would type Refgenie pull, and now you'd have the FASTA file for HG38. Or you could build something with Refgenie build. Once you've got an asset locally because you've either pulled it or built it, then you use Refgenie seek, and that provides you the path to the object. So Refgenie is sort of this middle interface where you now no longer have to worry about paths. All you have to do is have HG38, and then Refgenie will be able to identify those paths on any system. Next slide. So this is my last slide. How would you do this from within R? So the Refgenie client package is written in Python, and you can access it through R with the reticulate package. So this is something that I'm sort of just getting into and trying to figure out how it works and what's the best way to wrap this up. But essentially, you can just call this code like this, and from within R, you could pull assets from the server, or you can seek and identify local paths to assets, all from within R through this reticulate package. So I'm currently exploring the possibility of wrapping this up as a separate package and maybe making the interface a little bit easier from within R. And I'd be interested in talking to anyone if they're interested in that. Last slide. That's it. The last slide's just a few links. If you want to check it out, please do so or contact me later. I'm done.
Thank you very much, Nathan. Do you like to add these links also to, I don't know, to the platform perhaps?
Sure, or I'll put just one of these links on the chat if someone wants to follow it. You can get to all the other things from one of them.
All right, that's very helpful. Thanks very much. The next speaker is Antonio Colaprico. I hope this is the correct pronunciation. Let me see. Here we go. So, all right. Hi, Antonio.
Hi. How are you?
Good, thanks. And you?
I'm good. Thank you for this opportunity. So I'm Antonio Colaprico. I'm an associate scientist at the University of Miami. And today I will present our tool that we call the Moonlight, to do interpret pathway to discover cancer-driven genes. Next slide, please. So first of all, we made also this poster. I had this link in the 1000 I presented yesterday. And we had the opportunity last year in the Bioinformatician 2019 to show, I made a workshop, and I had the link here, the PLC workshop 2019, where I presented not only Moonlight, but other tools that we developed. Maybe you know my tool TCGA links to retrieve data from GTC and TCGA, cancer subtypes, vitamin, and so on. So what's novelty in the Moonlight package that we published recently this year in Nature Communication, please can you go next slide, it's an integrative analysis. So what we want to do is to discover driver genes, so it means oncogenes and tumor suppressor using multi-omics. So the hypothesis that we made is that we can consider oncogene using a network approach, that where genes that increase proliferation of cells or decrease apoptosis, they can be oncogene. Whereas genes that decrease proliferation of cells but increase apoptosis, they can be tumor suppressor and other cancer-associated biological processes. And then also, as you see in figure 1 here of the paper, we used multiple layers to understand mechanism of activation or inactivation of those oncogenes in tumor suppressor. For example, we looked at DNA methylation, and we see it in this B. So we found hypomethylation of oncogene and hypermethylation of oncogene, of tumor suppressor, sorry. And we found amplification of oncogene and deletion of tumor suppressor. I also showed this copy number reshaping landscape in a manual curation, but we had also a machine learning approach, so we could train Moonlight on already validated cancer driver genes to give weight of those biological processes. We also used chromatin accessibility data. So like the figure A is that we can perform like a function enrichment analysis. So looking at the vignette and bottom, but if you have just a list of differential space genes, and you want to see those enrichment of biological processes. Can you go next slide, please? So what we discovered that as a novel findings that so those genes that increase proliferation and decrease apoptosis in two manners, we call it oncogenic mediators. That we discovered this in somatic and promoter intermutation. So for example, we found association here in figure 3 of oncogenes in promoter mutation, but increasing of tumor suppressor in intron mutation. And when we look at chromatin accessibility, data and ataxic data from TCGX, so the recent data published in the science paper. So we found oncogenes that are associated with the open chromatin in promoter region, and instead we found tumor space associated with the open chromatin region in intron region. So there was also a recent published science paper that confirmed our finding that we said that it's the tissue type specificity that allow the permeability or the epigenetic background, the tissue that allow a gene to change role. So if you go on slide.
And that's time too. You want to wrap up?
Okay, the last slide is for, I can wrap up. So we used recently in those two set papers to identify oncogenic process associated in somatic and germline comparison, frame shift and nonsense mutation, and also oncogenics and the differentiation associated with the stamens. Last slide, I want to thank everybody that contributes, not only me, and the Back on Data 2020 for the Travel Award Scholarship, and everybody for the attending. If you have a question, you can write me. Thank you.
Thanks very much, Antonio. This was very good. Thanks a lot.
Thanks.
So I'm sure there will be questions on the platform. All right, the next speaker is Iguaracy Pinheiro-de-Sousa from São Paulo. Here we go.
Okay, I'm feeling a little bit outside here because I will present a bit more biology than bioinformatics, but okay, let's get started. I'm Iguaracy, PhD student at the University of São Paulo in Brazil, and I'm going to present a part of my PhD. And thank you, Bioconductor, for the invitation. You can go next. Well, what I'm trying to do is to identify a molecular signature for endothelial dysfunction. But first of all, what is endothelial dysfunction? Basically, it's a condition that precedes several of cardiovascular diseases. And some of the problem is a lot of risk factors can cause endothelial dysfunction. So based on that, I went to GeO database to identify the experiment that had these main influence factors that induce endothelial dysfunction. And we end up with 24 databases that we can see here. The main is abdominal flow, inflammation, lipids, and hypoxia. So what we did was to do differential expression analysis in each one of these conditions. And then we did in each one of the data sets, and then we did the meta-analysis for each one of these conditions. And we kept the genes that were differentially expressed in all conditions. Then we go to validation in human coronary artery endothelial cells and in ex vivo in human artery tissue. And you can go next. You can go next. Okay, thanks. We identified 12 genes differentially expressed in all conditions. And when we saw these results, we realized that it wasn't so straightforward that as we thought, because the genes vary according to the condition. But fortunately, one of those genes that's not 3 here, it's a very sensitive gene for endothelial dysfunction. So every time that the endothelial cells are under stress, this gene will be downregulated. And we can see here that's downregulated in all conditions, meaning that it's kind of a positive control of our analysis. The other problem in the GeO database is that hypoxia, inflammation, lipids, the 3 other columns, most of these experiments were done in static conditions, and the endothelial cells are always under flow condition, under the blood flow. So what we did was to put the endothelial cells under the blood flow to simulate. And here you can see in the right upstream part that we set up as background the fluid shear stress. So the fluid shear stress would be like a good blood flow, and the oscillatory shear stress would be the pathological one, the abnormal one. And we combine here, if you can see in the conditions, with the chemical factors, hypoxia, IL-1 beta, oxidized lipids, and all of them together. And what we can extract here as information is that, for example, some of the genes are more susceptible to some specific stimuli, for example, CXCR4, were more upregulated to oscillatory stress rather than the other stimuli, for example, when we compare it to fluid stress. But VEGF-C and FKB, IOA, are more expressive when you have the IOL-1 beta stimuli, so the inflammatory. And when we thought about the human condition, if you can go next, everything is happening at the same time. So we got human tissue from 30 donors, and we divided in three categories of lesions, going from the minor obstruction to the major obstruction. And we can see that some of those genes were actually increasing its expression according to the degree of the atherosclerotic lesion. So yes, we identified a molecular signature for endothelial dysfunction using public database, and we are able to validate in vitro and ex vivo. But the nicest thing is that some of those genes were never associated with endothelial dysfunction. So yeah, thank you. That was it.
Enough time.
I was afraid of running out of time, but thank you.
Thanks very much, Iguaracy. This was very nice, and also thanks for staying on time. I hope there are also questions for you later on. Now we'll move to the next speaker, and this is David Burton.
My name is David Burton. I'm a student in the McCall lab at the University of Rochester Biostats department. About some of the statistics behind an update we're making for the ternary net package. So I'm calling this mixture modeling for genetic regulatory networks from single perturbation gene expression experiments. Slide, please. Thanks. So some of the motivation here is that cancer genetics researchers believe that genes work together to grow tumors. One of our collaborators, Helene McMurray, was lead author on a paper where they felt they had sufficient evidence that the genes were working so strongly together that they should just call those genes their own group or give them a new classification, that the gene class was working so strongly that it was affecting how they define cancer. The next year, another group started looking at gene networks from a systems perspective, as if the gene network went into an attractor and stayed there, and that's what was causing cancer, or was part of cancer existing in the state, was that the gene network just couldn't get out of there. So the thing is, if we want to look at gene networks as a network, but we're using gene expression data, we kind of have to have some kind of a device or simplification to make that jump sometimes. Slide, please. I have a mixture model overlaid on some delta CT values from some single perturbation gene expression experiments. And so the simplification or assumption we want to make is that when the genes are overexpressed in response to the experiment, then we represent them with a uniform distribution in the positive space. When they're underexpressed, we represent them with a uniform distribution in the negative space. And we use a normal distribution in the middle for genes which have not been affected by the experiment. So if we use this simplification, that opens some new tools up to us. Slide, please. And what that does is it allows us to then calculate probabilities of those gene expression values based on which network state we say that they're in, whether they're up, down, or if they're baseline. And then we can calculate a likelihood for any possible network fit given that data set. Using that tool then of the likelihood, we have a parallel tempering scheme set up that samples possible networks from the space of all possible networks for the 20 genes that we're looking at. And then we can compare those network fits to one another based on which ones score the best likelihood. Slide, please. What that allows us to do then is look at the top scoring networks and see how many times or what proportion of those networks have the same gene relationships occurring in them. And so we get very strong results sometimes where virtually all of the networks that were scored well show a relationship, and sometimes not so strong. But this graphic here just kind of illustrates some of the output we're trying to get at with the new scoring method. And that's pretty much it. Slide, please. So I'd just like to thank my advisor, Matt McCall, the undergrads who've worked with me, Daniel Munoz-Huerta and Michael Lansford, our collaborators in biomedical genetics, Helene McMurray and Hartmut Land, and our computer support guy, Harry Stern. So thank you guys for your time.
Thanks very much, David. All right, so the last speaker is Aaron Chevalier.
Hi, I'm Aaron. I'm a PhD student in the Campbell Lab at BU in the Department of Bioinformatics. So I'm going to be talking about a new R package for mutational signature analysis. And I'm going to talk really briefly, do some background on mutational signatures, talk about the features of the package, and then walk through kind of an example workflow of use. If you could go to the next slide, please. So unlike some analyses that are interested in the effect of mutations in causing cancer, for instance, mutational signatures are looking at the causes of mutations. So whether they're environmental exposures or normal processes in the human body. So for instance, smoking, next slide. Benzoapyrene is a component of cigarette smoke that creates DNA adducts that create not random mutations, but specifically C to A transversions in the genome. So they have a specific signature. Next slide. So when we think about, yeah, and that's perfect. So cigarette smoke, we can actually show this as a probability distribution across different types of mutations. So we see those C to A transversions very highly, but also within that, different probabilities for different upstream and downstream mutations, which is what that's broken into. Next slide. So our package takes VCFs, MAFs, tables, and counts these mutations per sample. And from there, we can deconvolute these counts into signatures and exposures. So currently, we provide NMF and LDA for doing the deconvolution. So in the middle left, we have the signatures, and on the middle right, we have per sample, we have the proportion of the exposure to different types of mutations. And then we have downstream analyses for comparing unknown signatures to known signatures, cosmic, for instance. We can also break these exposures down by annotation, for instance, tumor type. And we can also, so we use a LDA posterior implementation for predicting exposures in new samples. So this can be at a cohort level or it could be a single sample. So VCF MAF for table, we support SNVs, so single nucleotide variants, and we can also calculate transcription strand and replication strand, double base substitutions, insertions, deletions, and also any custom annotation you may have on a region in the genome on a mutation. And we can also mix and match these and perform deconvolution for any kind of custom analysis you might want to do. Next, please. So here's our kind of example analysis. So we've got our exposures on the left, and there's probably some patterns in here, but we can't really see what's going on. And we have our signatures on the right that we can maybe guess what these are based on what our prior knowledge is, but as of this point, there's not much we know about it. Next slide, please. So using one of our analysis plots, we can see that there's something strange happening here where signatures 1, 2, and 4 have tumor types that highly represent them, but signature 3 is really just these kind of outliers. Next slide.
And that's time if you want to wrap up.
Okay, so there's a lot of analyses that we can do. We support UMAP as well, but yeah, if you could just go to the last slide. So we can see that there's just these few mutations that have very high mutational counts. Next slide. And we can see if we compare the signatures that the signature 3 that we're seeing here, if you go to the next slide, yeah, relates to cosmic signature 10, which is known for being a hypermutator, and it's found in these tumor types. So we're just refining things that we already know at this point, but these same tools can be used for novel analysis. So if you could go to the next slide. This is our GitHub for the package, so we're working on a manuscript now, and then we'll submit to Bioconductor. Thank you so much. Sorry for going over time.
No, that's fine. Thanks very much, Aaron. You are very welcome to perhaps copy this link into the platform so the people can also find it later today and tomorrow. This would be super nice in the platform under chat, for example. All right, so thanks very much to all speakers. And now we have time for questions. Thanks, Kyla. Let's have a look at the questions.
Sure. So I've copied over the ones from the polls. The first question is to Kristyna. Does your package provide facility to intersect multiple bed files to calculate the fraction of genomic feature overlaps?
Thank you for the question. Unfortunately, at this point, our package does not offer this feature, but it is definitely an interesting suggestion. So we might actually consider including that. So thank you for that suggestion.
Next question for Nathan. Did you look at the Basilisk package for interfacing with Python?
Yes. I just was actually looking at it a couple of days ago, and I sort of wrapped Refgenie up in both Basilisk and Reticulate. It seems to me like from my just initial look at it, it seems like what Basilisk is doing is essentially just wrapping Reticulate so that you have a version of Python that's sort of embedded. So it's really Reticulate under the hood is what it would be using with either Basilisk or Reticulate. So, so sure, it would work. What Basilisk would add is a self-contained version of Python, which, you know, it just depends on if that's what you're interested in.
And then there's a second question also for you, Nathan. Can Refgenie be used to pull from NCBI or Ensemble/GenCode? Illumina does not appear to have updated human gene models in iGenomes since 2015.
Yeah, I mean, the lack of updates in iGenomes was one of the reasons why we wanted to, you know, produce Refgenie. The lack of updates, and also just that they didn't have the particular, you know, spike in control genomes we were using. But anyway, to answer the question directly, I'm pasting in the chat now a link to the actual resources where you can go browse what's actually on there. It's basically whatever I put onto the server is what you would be able to download, and they're hosted on S3 in Amazon's public dataset program right now. But I can add, I guess, whatever. So if there's resources on there that people think would be useful for the community that I haven't put in there, I can add them. Right now, the human genome that we have is, I think, an NCBI genome. And we also have some GenCode and Ensemble annotations already on there. But if you have an idea for something that's missing that you think would be valuable, just let me know and we can figure out how to add it. Thanks for the questions.
So the next question is for Iguaracy. How did you score the severity of the lesions?
Well, if you have here the lumen of the vessels, and when you have a high atherosclerotic lesions, you probably reduce this lumen of the vessel. So you have a vessel more obstructed. So we have the less degree. So you have like the whole vessel, less obstruction to more obstruction. Probably you have a severe atherosclerotic, and that's how we graded all the atherosclerotic lesions. Thanks for the question.
Great. So the next question is for David. How do you infer the directionality of the arrows? Isn't the likelihood of both directions equivalent?
Actually, the direction of the arrows comes from the network topology that we sample. So I mentioned this very briefly, but there's a parallel tempering algorithm we run that samples networks, and the parent nodes in the network that we're sampling are randomly generated. So we don't actually directly infer which gene is a parent to which other gene, so much as after we're given a scenario, we then use the likelihood to score whether or not it's doing well, so to speak. As far as whether the likelihood of both directions are equivalent, setup it is where there's single perturbation experiments. So any genes that we're interested in, one of them gets moved either up or down and is kept there in the experimental condition, and then the other genes react to that gene being forced to overexpress or underexpress. So I wouldn't say that the likelihood of both directions is equivalent here. It's not exactly the same as if you're doing like what is it, co-expression. In that case, I think maybe both directions are equivalent, but I think because of the experimental setup, that's not really true here. I hope that was sufficient.
So the next question is for Aaron. Can you say a bit more on the statistical model that you use to identify the signatures?
Yeah, so for discovering new signatures, we use non-negative matrix factorization, which is standard in the field, and we also use latent Dirichlet allocation, which has been widely used for text mining and is starting to be a little more common in the mutational signature space. And also for prediction, so for taking known signatures and predicting exposure using those, we use an LDA posterior. And for all of these, you can input a seed so you can get reproducible results.
And then we have one final question that came in for Kristyna. Does GenomicDistributions do rainfall plots?
So thank you for that question. I had honestly no idea about what even rainfall plot is. So I was just like looking it up. So at this point, it's not part of the package, but when I looked it up, it shouldn't be like really that hard to implement. So like, I guess, again, thank you for the suggestion. I'm gonna write it down so we can maybe implement it in the future. But at this point, it does not allow this option. But on the other hand, like what I saw, it's just you need a position and the distance between the features, which are both calculated without genomic distributions. So again, like you can calculate these, and then you can, at this point, you can plot it yourself. But thank you for the suggestion.
Oh, wow. Thanks very much to everyone again. We still have time for discussions. Are there more questions from the participants?
Actually, I have a question.
Yes, please.
What I want to know if Antonio is using any kind of causality approach in its integrative analysis for the genes that he's finding.
I don't know how to pronounce your name. Iguaracy.
Iguaracy, but that's fine.
Thanks for your question. I think it's a good question. We are not using yet some causality. Those are important. I mean, for example, for the mRNA degradation to identify microRNA that they can have oncogenic role, but we are planning to do in the future. If you have suggestion or want to collaborate, you can send us an email. Thanks.
More questions, ideas, suggestions? All right, then yeah, so thanks very much to all speakers again. Thanks, Kyla. Thanks for everyone for joining this session. We will close a couple of minutes earlier, so you have a longer break and before you join the next session, I guess. Have a nice day, and see you later today and tomorrow at the conference. Bye-bye, everyone.